Fourth lecture in series on machine learning for biochemical fourth lecture in series on machine learning for biochemical applications. I'm Devansh Pandey. I'm a second year PhD student in Dr. Narasimhan's lab, and today I'm going to be talking about embeddings and transfer learning. So before we go ahead, let's first revisit the cat model that we saw in first lecture that was taught by Clay. So in this model, our inputs are images. Those images could be of cats or different objects which are not cats. And our model tries to learn to identify whether the input image is a cat or not. So what does this model doing here? This model is learning the features that are relevant for identifying whether something is a cat or not. So it might learn features like whether the object in the image have whiskers, whether they have pointed ears, whether they have a tail, how the paws look like. That's what the model is learning. And these learned representations are saved in the model, are stored in the model as vectors, which are called embeddings. So if a model learned 10 different features from your input data set, then that model would have a 10-dimensional embedding. And those embeddings are stored as vectors. So let's first go over this example. So here what we are trying to do, here we are inputting an image in the model and we are asking that model to reconstruct that image. So how the model is doing that, first you provide it a input image and then model is encoding that image into a vector. That vector is basically the embedding. So the model is looking at the image, it is identifying the different patterns in the image where the pixels are black, where the pixels are white, and it is learning something that it is storing in this compressed representation, which we call the embedding. Now this embedding is getting passed into another component of the model that is called decoder. And using those embeddings, that decoder is trying to reconstruct the input image that it was given. But here you can see the original input and the reconstructed input are slightly different from each other, right? They are not exactly the same. So that means the model is only trying to learn what is really important in reconstructing this input, but not on the very minor details. So that's why you see the reconstructed input is a bit smudgy. And so what are the key features that are learned in the embeddings? So first of all, embeddings contains the most relevant features that are required to differentiate between all forms of input to the model. The second other important thing about embeddings is that these are of constant shapes. If we are working in images, most of your data would be of constant shape. That is like the width and the height of the image is constant. But suppose you're working with amino acids, right? Your amino acids could be of different lengths. But when you convert them to embeddings, all the dimensions are same, and it is easier for these neural networks to learn something that is of a constant shape as compared to something that is of different shapes. And the third thing that is happening that is getting stored in this embeddings is the features. That is, you could just pass the amino acids, a large amount of different varying amino acids to the model, and the model will be able to learn general features about the amino acids without you explicitly telling anything about it to the model. So that's are the key features that are stored in these embeddings. So to illustrate more, I'll go over this example, and I chose this example because I learned embeddings using examples like that. Embeddings make more sense when you think of them in context of words or the words in English. So suppose you consider this model where your input is a text in English, and this is just a toy model. That's not the actual model that does this translation. And you pass it to the model, and the model's task is to convert that into German. And during that process, what the model will learn is different semantic and syntactic relationships that exist in English language. And those relationships would be stored in the word embedding vectors. So how do you get that word embedding vector? So what you can do is that instead of making a prediction, you can cut the model before the last few layers, and you can take the output, and that output would represent the vector that has learned things about the syntactic and semantic relationships in the English language. So to illustrate, so how do they capture these relationships? So let's consider this example here. So suppose we have like a two-dimensional vector and we plot it on like a two-dimensional plane. So this vector represents the vector that is learned for the word man, and the other one represents the vector that is learned for the word woman, right? And now, since these are vectors, you can perform mathematical operations on them. So if you subtract the vector, if you take the vector for woman and then subtract the vector for men, or you calculate the Euclidean distance between the two vectors, you will get a distance. And then you take two other examples, that is the vector for a king and the vector for a queen, and then you again calculate the Euclidean distance, you will get a value. And when you compare both of these values, they would be quite similar to each other. That means it is trying to learn semantic relationships. And if you go to the left panel, you can see it also learns syntactic relationships. So for the first one, suppose you take the vector for walking, and then you subtract the vector for walk and add the vector for running, you will get a vector that is approximately similar to the vector that looks like running. And this is true for all the different syntactic relationships in English language. So here I have talked about plotting it in like 2 dimensions. That is like our vector only have like two dimensions, X and Y. But in reality, if you're training a big model, it will, yeah. This one. Yeah. Yeah. Okay. Oh, okay, I'll fix that. Um, yeah. Mhm. Okay. Hmm. Hmm. Okay. Okay. Thanks for pointing it out. Um, okay, so before we are only like working with 2 dimensions, but these big models, they try to learn a lot of different features. So in this example that we are showing here, so this is a 3-dimensional space. That is, the vector has like 300 different elements. And when you like plot them in the 3-dimensional space, each of these points would represent a word, right? And suppose you take a query vector for kitten, and you look for the space around the query vector. So the things that you will find is that near the word kitten in the space, you will find the word for cat. And you will also find things like dogs, wolves, chicken. So it is, what it is saying is that it is grouping things that mean, it is grouping things together that mean the same. So it is grouping animals and pets together. But if you go to the other side, you can see it is also grouping fruits together. But there is this one caveat, that it is grouping Apple, the corporation, with Apple, the fruit. And why is it doing that? Because this model learns these vectors in a context specific way. So sometimes it gets confused, and then it brings things together that are not that does not mean actually the same. So, but one question is, could this only be applied to words? What about, can we get embedding vectors for different biological data sets, or like proteins or amino acids or DNA sequences? I think this is the second time you guys are seeing this image. It was first shown by Clay in the lecture 1. So this here, what I'm showing is that these are the embeddings learned from a protein language model called ESM. And when these embeddings are plotted into the space, they look something like this. So would anyone want to tell me what are the things that are stored in these protein embeddings? Like in words, it was like syntactic and semantic meanings of the words in English language. But when you look at like molecules or proteins or DNA sequence, what kind of information would be stored in these embeddings? Yeah. Mm-hmm. Yeah. Yeah. Anything else? Yeah, it can. Mm-hmm. Yeah. So here they learned like the structural and functional relationships between like protein sequences. So here when you like plot these embeddings, you could see like the proteins that have similar function will group together in this embedding space. The proteins which are hydrophobic would group together. The proteins which have like the same secondary structure will group together. So it is trying to learn the biologically relevant features from the data. So we can actually generate embeddings for every possible input to a neural network. So if you train a protein language model on amino acid sequences, you can get them embeddings for those amino acid sequences. One other example is computer vision model. These are the models that take images as input. So if you take a computer vision model that is trained on identifying human posture, you can the embeddings that it learn can learn things about basic human skeletal form. So let me go into the examples. So suppose you have this data of amino acid sequences and you run them through a protein language model, and you can generate embeddings for all those amino acid sequences that you have in input. So now using these embeddings, you can either perform classification or you can perform regression. So if you want to do classification, so what you can do is that take these amino acid sequences, generate embeddings, and then run those embeddings through a simple logistic regression model. And using that classification model, you can predict things like gene ontology or fold or secondary structure, all the classification tasks that you can do on amino acids. And if you want to do a regression task, you can again run those embeddings through a linear regression model, and you can identify the isoelectric point of the amino acid sequence or the Gibbs free energy or other regression tasks that you can do on amino acids. So one question is, why is it better to use these embeddings as compared to raw input? So this protein language model, the one that I show here, it is trained on diverse amount of amino acid sequences, and it is trained on really large data sets. So the intuition behind training it on such big data set is that it learned biologically relevant features about amino acid sequences and about their homology and coevolution and other stuff. So instead of just training on raw input, what you can do is that you can get the embeddings from the pre-trained models that will give you more informed features about your amino acids as compared to building the feature yourselves or building a big model yourselves to learn those features. So you already have a good informed guess in the form of these amino acid embeddings. The other thing is the constant shape, which I talked about. So if you have a constant shape and good features, you can use simple models like logistic regression and linear regression to predict things that are not very simple, things that are complex. And again, these embeddings contain relevant features about the molecules or the protein sequences. Okay, let's look at the next example, the one which I talked about earlier. That is, if you take a model that is trained on either images or videos to identify human posture, right? So this model takes input as an image or a video, and that input is processed through the model and it produces an output. So if you could see here in this video, so like this model is able to identify the postures of different people walking and dancing in this video. So now you have trained this model, and this model has stored the knowledge of identifying different parts of the body and different postures in humans. So what you can do is that you can use this model, you can use the knowledge of this model to do something else. So here what I show is that we have whole body DEXA scan images of people. And then when you pass these images to the model, you can do things like identifying different bones in the body or identifying the distance between the different bones, like shoulder width, hip width, identifying the length of the bone. So see, this model was not trained on these kind of images. It was trained only on identifying poses in human body. But during that training, it is learning something about basic skeletal form in humans. And you can, again, like just use the knowledge of that model and predict these kind of things. So that's where embeddings and knowledge from the models come into play. Does anybody have any questions on embeddings before we move? Yeah. This one? Yeah. Yeah, but Okay. So the data that it is trained on, like images and video inputs, they have like collected them throughout the internet. So we have like a large amount of data set. But whereas if you go here, we have like a limited data set, right? So a model that is trained on large and diverse data set would learn generalizable features that are really important in distinguishing this to distinguish between different forms of input. But if you just train on these, you can do that if you have like good amount of data set and your model would be good. But when you do not, you take help from the models that are trained on big data set, and then you like take those model and adjust them in a way that you want to use them in. Yeah. 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 So that, yeah, like here we are identifying different landmarks on your body, right? But when you ask the question, what are so suppose you pass your data through the model and you learn like a three-dimensional embedding, right? Every like it is a vector with 300 different dimensions, so it is very difficult to answer what those specific 300 dimensions are learning. The only way to identify those is by like plotting it and seeing like what kind of things are clustering together, what kind of words are clustering together, what kind of protein sequences are clustering together. Because since like now like you have like taken a word and then converted into something like multi-dimensional vector, so like you lose some explainability, but but you believe that that vector has learned something relevant to your input. Yeah. Mhm. That's like the best. Mhm. 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 This one? Mm, it is similar across different data modalities. Like if you are giving images, right, it will try to learn the like local patterns in the images and the global pattern in them. You just like convolute over the images over and over until you get to the point where you feel like, oh, I have learned enough things, now I can make predictions. Yeah. So like convolutions across different images, if you're like training on cat images, or if you're like training on X-ray images, it tries to do the same thing, same mathematical operations. Yeah, you don't change those. Any more questions? Okay, great. I think we had like really good discussions on embeddings, and I think that helps clear things up. Yeah. Okay, now I'll move on to the next section that we have is transfer learning. So what is transfer learning? To understand that, let's first look at what is traditional machine learning. So in traditional machine learning, you make a model for each task. You make a different model for each different tasks. And that does not matter. So suppose you have like same amino acid sequences, right? But for all those amino acid sequences, you have like different properties, right? You have, suppose you have like 1,000 sequences, and for all 1,000 sequences, you have isoelectric point, you have your secondary structure, you have your Gibbs free energy. So for to predict every diff every different things, you have to train different models. That is traditional machine learning, right? But that at some point is really difficult. So for each different task, you have to do that work every time to predict a different thing on the same data. But instead, what people have figured out that you can do is called transfer learning. Because since your input data is kind of similar, right? If you look at that example of HRNet model, the input was images of people, right? And our test data was X-ray images of people. They are kind of same. So instead of training a different model for each of these different images, what we can do is that train a big model and then store its knowledge and then transfer it knowledge for data set that looks similar to the data that you started with, right? So this is like a really good example of transfer learning. Like you take a model that is trained on different kind of data set, adjust it to your needs to do the things that you want to do. So before moving forward, I wanted to like discuss a few things because these terms will come up in the next few slides. So in machine learning, we have different training paradigms. So the first, and all of these paradigms are solely dependent on the type of data that you have. So if you have labeled data that you have like your and dependent variables, the learning that you do is supervised learning. It is called supervised because you're telling the model, my input looks like this, and using that input, I want this output. So you are supervising the model. And the task applications of supervised learning is classification and regression, right? And then in unlabeled data, you can do two different things. So you have unlabeled data. That is, you just have your DNA sequences or your protein sequences, but you don't have any quantifiable or like a task that you can task data that you want to do on these sequences. You just have a large amount of sequences. So there you just have the data, but no labels for those data. So in that case, you can either perform self-supervised learning. What does that mean? So suppose what we were talking about before, ChatGPT. ChatGPT is trained on self-supervised learning training paradigm. So what ChatGPT does is like you give it like a text sequence and you cut it, suppose like you give it like a sentence with like 10 words, you cut it at 8 words and ask ChatGPT to predict the next two. So that is called self-supervised learning. You can do the same for amino acid sequences. You give it an amino acid sequences, you remove the last few amino acid sequences and ask the model to tell you what those amino acid sequences could be. Or you could also do fill in the blanks. That is, you take amino acid sequences, you randomly remove, let's say, 5 amino acids and ask the model what those 5 amino acid sequences are. So this type of training is called self-supervised learning. And the last one is unsupervised learning. Here you also have unlabeled data, but instead of doing the tasks in which we did in self-supervised learning, here we are doing more abstract tasks. That is, we give the model the data and ask the model to cluster it. Like you just give it amino acids sequences and ask the model to cluster them and see how those clusters look like, whether those cluster contains the functional relationships or the structural relationships in the model. And you could also do it for dimensionality reduction. So suppose you have this unlabeled data and your model has learned like 300 dimensional embeddings, right? But to perform certain tasks, all those embeddings would not be important. So what you could just do using unsupervised learning is that you can just pick out the features that are important for your task and use them. Does everybody understand? Yeah. So one example is PCA, which I think a lot of people use. So in PCA, what you do is that you select the dimension that explains the most variance. So suppose you are like given with the data set that has like 100 different features, and you want to perform a particular, perform a particular task, right? So what would happen is that not all of the 100 features would be really important in predicting something, right? So using techniques of like dimensionality reduction, you only retain the feature that are really important in performing your task, right? Any more questions? So in transfer learning, we have two different things. The first one is called pre-training and the other one is fine-tuning. So what does pre-training mean? So let's talk in terms of how a GPT model is trained. So pre-training in GPT model is that you give it sequences and you ask it to predict the next in sequence tokens or words, right? So in pre-training, the purpose of pre-training is to learn general features from large data sets. And the training data here is unlabeled. So as we discussed before, when the training data is unlabeled and you try to do a task where you predict next in sequence or you predict the mass sequences, the learning objective here is self-supervised learning. And during this process, your model learns generalized features about the data set. And it does not learn something very specific, but like general features. But what you can do is that once this model is pre-trained, it has the knowledge about the data set. You can take that pre-trained model and you can just adjust that model's weights according to your needs. And that part is called fine-tuning. So in fine-tuning, we make our model adapt to the specific task that we want to perform. And for fine-tuning, we have task-specific labeled data. And since we have labeled data, fine-tuning is supervised learning. And in pre-training, since we do not have labeled data, the task is self-supervised learning. So let me give you an example. So this paper came out this January. And in this paper, what they try to do is that they have input DNA sequences. They first convert them into tokens that model can understand. So here the input was like a 6,000 base pair long nucleotide sequence, and the researchers, they like chop it up in like 6-mer pairs. So you can imagine the model would have 1,000 tokens now, right? So out of those 1,000 tokens, the people they masked 10% of the token. So they masked 100 random tokens, and they wanted their model to predict what those masked tokens were, right? So they trained a deep learning model called transformer, and we will go to more details about transformer in the upcoming lectures. Just for now, you can think of transformer as something that is filling in the blanks here, that is predicting what the mask sequence would look like. So what this transformer do, for every token, it will generate an embedding. And if you put that embedding into language model head, you can predict what those mask sequences are. So here in your pre-training, you are just trying to fill in the blanks in DNA sequences, right? So during this pre-training, we believe this model will learn certain general purpose features about DNA, how different DNA elements look like, how promoters look like, how enhancers look like, what does a coding sequence mean. That are the kind of things that we believe this model will learn during this pre-training. So once you have this pre-trained model, and you can take the model's knowledge and use fine-tuning to do your tasks that you want to do. So here we are taking the model that is trained on predicting the blanks in DNA sequences, and we are fine-tuning this model to identify whether an input DNA sequence is a promoter or not. So here you do the same thing. You input your DNA sequence, and you tokenize your input. And so when you see this figure, it says like rescaling weights. So what does that mean is you take the pre-trained model and you adjust it minutely such that it gives you the best result for your task. You're not like making any drastic changes in the model. You're not adding any more layers, but you are just like fine tuning it and readjusting its weight to suit your task. Now you can generate the embeddings, and on those embeddings you can perform classification, and that classification could provide you probability whether the input was a promoter or not a promoter. Does anybody know what classification model we can use on these embeddings? Yeah, we did it on amino acid embeddings, right? The example that I showed you before. That is, we have the amino acid embeddings, and on those amino acid embeddings, we apply logistic regression to classify things like gene ontology or fold. You can do the same thing on these DNA embeddings. On these DNA, you can pass these DNA embeddings to a logistic regression and ask it to predict the probability whether the input was a promoter or not a promoter. So here it predicts that it is 85% sure that the input sequence was a promoter, right? So this do people now have a good general idea what pre-training is, what fine-tuning is? Right. Any questions here? Yeah. Tokenization. So there are like different methods to do tokenization. Here they have just performed a very simple tokenization. Since we are dealing with DNA, right? So the best way to chop up DNA is like in different k-mers. So you can chop it at like either every third position, fourth position, fifth position. So you have like different k-mers. So here they have 6 mers. So that is, they have chopped DNA at every 6th position. Yeah, they've tried different tokenization schemes. Like they tried taking 3 mers, 4 mers, up till 8 mers. But what they observed here is that 6-mer tokens were giving the best performance. So they kept that in their final model. Right. Yeah. Mm-hmm. Uh-huh. Yeah. Yeah. But though those codon only matter inside the coding sequence. But since here we are like training on the whole genome. So at some places, 3-mer would not be the best tokenization scheme to predict something. That could be a good tokenization scheme if you're trying to predict whether something is a coding sequence or not a coding sequence, whether like this particular piece of DNA will code for a gene and will not code for a gene. Their 3-mer tokens could be better, but here they have tried like all different strategies. They have tried like 3-mer tokens, 4-mer tokens, 3-mer tokens with like different strides, and they found out that like 6-mer tokens with like a stride of 1 were working very well. So stride, yeah, so like stride of, sorry, stride of 6-mer token with stride of 6, but they've tried 6-mer token with stride of 4, so they have like two nucleotides overlapping. They've tried 8-mer tokens with stride of 5, but the best that were working was like 6-mer token with a stride of 6. Yeah, no overlap. Yeah. Mm-hmm. How do we do the rescaling? So what we can do is that, suppose you have this transformer model with like 12 layers, right? So the way you perform rescaling is that you do not touch the last 10 layers. You do not touch the first 10 layers. You just like take the last 2 layers, unfreeze them, and then fine tune the model, fine tune those last 2 layers. So like you again like so like when we start, our model has like, so when we actually start training during a pre-training, our model has like randomized weights, right? But that model adjusted weights according to the data that we provided. So every different layer has different weights. So this was a BERT model. The BERT model is something that predicts the mass sequence. That is, it fills in the blanks, right? So what they did is that they trained like a 16-layer BERT model during their pre-training, and then when they went to fine tuning for this particular task, they unfreeze the last 2 layers. That is, the weights of those last 2 layers could change, but the weights of the rest of the layers will not change. So that's how they readjust that's how the weights could be readjusted during fine tuning process. Mm-hmm. Mm-hmm. Yeah, you can say that. Yeah. Yeah. If they share similarity, the model performance would be better. If they do not, you can test it whether it performs better or not, but you can't say anything before you fine tune it on your data set. Yeah. 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 Same type. Yeah. 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 Mm. Okay, let's take the last example for uh, uh, transfer learning and pre-training and fine-tuning. So before this, we were like going through an example of DNA, right? Now let's look through protein. So why do we want to pre-train protein models? So during pre-training, what does the model do? It learns complex features. So if you provide it like a diverse, really big data set of protein sequences, it will learn different properties about those protein sequences and complex patterns that we could not like comprehend. That were not very explicit to say. It could learn implicit relationships between the protein sequences, right? And we hope during that process it also captures some biologically relevant features. And the third thing is pre-training is important because most of the times you do not have large quantity of good labeled data set. Most of the time you would just have protein sequences, but you won't have like any Y value for those protein sequences, any dependent variables for those protein sequences. So it makes sense just to like let the model learn general features of proteins. And third, like the the most important purpose of pre-training is knowledge transfer. That is, during your pre-training, your model will learn something about amino acids and proteins that you can use later on for your specific tasks. And then why do we fine tune? So the most important purpose of fine tuning is task adaptation, right? So during your pre-training, your model is either trained on predicting the next amino acid or predicting the amino acids that were missing, right? But suppose your task is to predict the isoelectric point. So everything that the model has learned while predicting next in sequence or predicting the missing amino acid would not be important to predicting the isoelectric point of that amino acid sequence. So you take that model and you fine tune it on your data set that serves the best for your needs, right? And you could also call it specialized learning. Your model is getting fine tuned on a specialized data set for which you have like high quality label data. And most of the times, like suppose if you work in a wet lab, right? Suppose you work like really hard for like 2 years and you have like data on like 5,000 proteins, but that data is really small for building a good deep learning model, right? So what you can do is take something that is pre-trained on big amino acid or protein database and then just fine tune it for your purposes. And then the last one is improved task performance. So there have been like a lot of studies in which they observe that if you fine tune a big language model on your task, it performs better as compared to if you build your own model by yourself. So in most of the cases, this fine tuned model would have really good performance as compared to something that you will build. Yeah. Does anybody have any questions? What's like why this will perform better? Yeah. Yeah, it's not the same task, but the hope here is that we learn something that is like very fundamental to that data set. Mm-hmm. Yeah. Mm-hmm. Mm-hmm. Yeah. No, you also update weights in transfer learning. In transfer learning, you can choose what you want to do, but in transfer learning, you can also update weights. So I think the model that I show here, the last few layers were like were updated, like the weights for the last few layers were updated on this X-ray images to give like the better performance. Yeah. So you can choose to update the weights or not, but in fine tuning you surely updates the weight. In transfer learning you can do both. Mhm. Yeah, and I think that is all for today's lecture. I hope like everybody learned something about embeddings and transfer learning.